Hello guys, hope you're all doing well. Myself Vishwant. Today my topic is about biosafety regulations. Introduction. As you guys know, what is a biosafety? Biosafety refers to the safety practices that are to be followed on the laboratories to prevent from disasters and contamination to the public resource and the public areas. As you guys know, there are lots of diseases spreading through the leakages from the biolabs. So in order to prevent that, there are some certain criteria that the laboratory should follow to prevent from our safety environment and safety research. So basically biosafeties have four levels. So based on the organisms or the research that you are doing, the biosafety level differs. So biosafety level 1 have the minimal hazard, that is the non-pathogenic bacteria or something that doesn't affect the other living organisms or human beings, or comes under the biosafety level 1. Biosafety level 2 or moderate hazardous, that can be a mild pathogenic diseases that causes to the humans. So it affects, but it doesn't come under a dangerous situations. Biosafety level 3 has a high hazardous, so it may deal with some serious viruses or bacteria or some protozoa that causes heavy dangerous to including the living organism and the humans. Biosafety level 4 is extremely hazardous, like this COVID-19 which spread during our pandemic days, or comes under the biosafety level 4. So biosafety practices, so in order to prevent from contamination, so we need to have a biosafety practices. So it includes personal protective equipment. It's like gloves and the suits that are wearing by the scientists and the researchers that they are using during their experiments so basically, for example, if the peoples are working on biosafety level 4, they will wear a particular laboratory coats that prevents them from external environment, and they provide sustain oxygen that is required to work. Safe handling and disposal of biological materials. While handling that pathogenic bacteria or viruses, we need to be very careful, as it may contaminate very easily. And while disposing, we should properly dispose, and we should not dispose them in a local areas. After a proper experiment, the disposal should be done carefully. Decontamination procedures. As the viruses have the vitality to transfer from one host to another host, so we need to sterile the viruses before we are decontaminating. So the risk involving in this, or it may be easily transferred from one other to another, so there are various consequences. Regulatory framework. So what's a regulatory framework? So there are some certain protocols and regulations to be followed by the scientists and researchers who are working under those hazardous environment. So the regulatory framework differs from country to country, and the major regulations are determined by World Health Organizations CDC, that means Center of Disease Control and Prevention, and majorly in India it is followed by Ibsc, that is Institutional Biosafety Committee. The training and proper educations are required for the researchers, so these peoples can follow the particular criteria to prevent from hazardous or dangerous incidents that are not to be happened. So the proper equipments are to be provided to the scientists who are all working under that particular environment, that is PPE, proper protective equipment. Challenges in the bios. What are the major challenges that can become under the biosafety? It's like the particular protocols need to be followed. Even a slight accident may cause a huge disasters that can affect the common peoples, and it can be easily contaminated. So that international collaborations, it's like dealing their virus from one country to another country, but while transporting this virus from here to here, the virus need a particular environment or the particular protocols that will prevent it from contamination. But while transferring from one country to another country, the proper protection may not be controlled, because during the transportation whatever can happen. so it can be easily spreaded from one area to another. So while dealing with that malicious or biological organisms, we need to be very careful, and we need to be maintain that per a particular protocol, that so that the contamination can be avoided. So Institutional Biosafety Committee, what is IBSC? So while, as we know, the biosafety deals with researchers who are doing genetically modified organisms or hazardous organisms. In order to follow that, so IBSC comes under Department of Biotechnology in India. The IBSC deals with researchers who are all dealing under that harmful viruses or pathogens. So they collaborate with other scientists and other field researchers so that they can gather the knowledge on the researches that the peoples are doing, so the major accidents can be prevented, and also to follow the biosafety guidelines. So what are all the composition that comes under the IBSC? So the various members are there in this biosafety commissions. So the different types of scientists and geneticists are, as well as safety officers, are responsible for making the biosafety protocol, so that the researchers can follow the rules and regulations, and legal advisors and every federal laws are to be followed, and they all join together and create the biosafety commissions. So the decisions are made together in order to have the knowledge that can given by each and everyone, so that it can be very useful and the decisions can be taken. Responsibilities of IBCs. Know the responsibilities that the IBC can, so whatever the biosafety committee, that is, it includes the IBC, the rules and regulations that to be followed while handling that chemicals and handling those microorganisms or viruses, whatever it may be. So in order to prevent from disasters, these biosafety commissions are used for the safety, and for each and every steps are to become under IBSC, so that it will not affect the peoples, and also to educate the researchers to follow the protocols, to do the experiments successfully without any dangerous activities. So what is the importance of IBCs? So as we know, if suppose let us think the IBC is absent, some researchers may not have the awareness about the things they are handling with, or suppose some scholars or students who are all dealing with the harmful hazardous viruses, they need to be follow the protocols. So IBCs are very important to do an healthy experiment, I mean to do an experiment without any contaminations or everything. So the risk management will be reduced, and to increase the safety more. Conclusion: overall, as we know, biosafety commissions play a major role on helping our environment to be healthy, and also for the researchers, so that the future generations can also maintain the particular orders to be followed. Thank you guys. I hope you understand the topic under IBSC.